If you can't hear me, please uh, please leave and rejoin the meeting. For the chapters we're covering today, we're covering chapter 3. So we are done now with chapter 1, with chapter 4. Our first set of quizzes is are gonna be covering chapters 1, 2, 3, and 4. So we're done with 1 and 4. Today we are starting our discussion of chapter 3, and this is going to be our first module. So chapter 4, the tissue part, as we've mentioned, the tissues we don't have to cover for the lab exam, the glandular epithelium, or the tissue repair. Those are the two topics that we have discussed at the end of last meeting, but they are not covered in the lab exam. So chapter 4, all chapter 4 is going to be covered by the lecture exam, but the lab exam only going to be covering the tissues part, but it's not going to be covering glandular epithelium or the tissue repair topic. For lecture exam, so this is the lab exam. Lecture exam is going to be covering chapters 1, 3, and 4 for now. I'm not sure, plus or minus 2 according to our performance. So lecture exam and lab exam are available for you to complete 1 week before the due date. Right? Both lecture exams and lab exams are going to be available for you 1 week before the due date. So I believe our due date for those quizzes and exams is going to be June 25th. So 1 week before, it's going to be June 18th. So expect those exams, lecture and lab exams, to be available by June 18th. So lab exam only covers chapter 4, but not the glandular epithelium and not the tissue repair. And both topics are in chapter 4. The rest of chapter 4 is going to be under the lab exam. All chapter 4 altogether is going to be tested in a different way in the lecture exam. So what's the difference between the lab exam and the lecture exam? The lab exam, as we've mentioned, it's going to be diagram based. Please use the visible body assignment for the tissues to practice on how the lab exam going to look like. Also check on Canvas under the lab resources. You will find a set of sample questions for the tissues lab exam. All right, so use those two as a guide of what you should be preparing for and how the questions going to be addressed for the lab exam. For the lecture exam, it's going to be similar to the Q banks. They are multiple choice questions, or true and false questions, similar to Q-banks, similar to your quizzes, your chapter quizzes. So make sure before you complete the lecture exam that you study the Q-banks or to use them as your study guides, and to complete the quizzes as a practice for you before taking the lecture exam. So again, both lecture exam and lab exam are gonna be available 1 week before the due date, and this is gonna be applied for all the rest of labs and lecture exams. This specific one, the first lecture exam and first lab exam is gonna be available 1 week before the due date. So the due date is June 25th, so it's gonna be available on June 18th. All right, does this answer your question? Anybody is confused about this? Any questions? Any questions here? All right, great. So today we are starting chapter 3, in which we are studying the cells. First, we're gonna be discussing an overview of the cells. We're gonna be spending some time discussing the structure of the plasma membrane, which is the boundary of the cell, its chemical structure, its interaction. Also, we're going to study the membrane transport, different types of membrane transport, and this is a very important topic. If you are taking the HESI, if you're taking the TEAS exam, if you are taking advanced biology later on, if you're taking pharmacology, if you're taking almost everything, anything related to science and biology, you're gonna need to know membrane transport. It's very important topic. One of the most important topics in this semester. One of the most important topics in this semester. You won't be able to understand the transmission of electrical signals in the excitable tissue like the muscle nerve without knowing how the membrane allows the transport of different things. You will not be able to understand the kidney physiology if you do not understand the membrane transport. It's very important to have a very good understanding on how stuff pass in and out through the plasma membranes. This is a critical topic, highly important. We're going to also discuss the organelles of the cells. Organelles. We're going to discuss few information about the nucleus and the cell cycle, which is the life cycle of the cell. We're going to discuss how the cells are going to be performing their mitotic division or mitosis, and this will allow them to divide. So again, very important chapter in terms of physiology. You're going to rely on this information a lot in your upcoming classes and later in this course. So let's start first with an overview of the cell. So we have a general structure of the eukaryotic cells, and this is the type of cells that we do have in our body. Not all the cells gonna be identical, but we're gonna be studying the general structure, the common structure of all cells. We do have different types and shapes and functions of the cells. Like for example, we've got some cells that have the ability to carry oxygen, like the erythrocytes. We've got some cells that have the ability to build up fibrous connective tissue. like the fibroblasts. Okay, they can build collagen fibers, they can build elastin fibers. I have cells which are going to be responsible for secretion of mucus, for example, like the goblet cells in the epithelium. We have epitheliums that will be responsible to form the body coverings and linings. We have cells that have the ability to contract, to reduce their length. Those are my muscle cells. And if you remember, we've seen three types of muscle cells forming the three types of muscle tissue. We've got skeletal muscle fibers, smooth muscle cells, and the cardiac muscle cells. We've got some cells that have the ability to store fat in the adipose connective tissue. As you remember, those are definitely going to be different from the white blood cells that have the ability to engulf bacteria, to eat bacteria, which are the macrophages, which are different than the nerve cells, which have the ability to conduct and create electrical impulses that will allow them to control other organ cells. So we've seen many different types of tissues, and each type of tissue has its own specific type of cells. So I have a lot of cell diversity in the body. I have cells that contain only half of the genetic material. They are highly specialized. They are responsible for reproduction. So we've got cells responsible for reproduction, like sperms, like the eggs. Those are specialized cells. I cannot reproduce using nerve cells, for example. So they need to be sperms and eggs. What is the generalized eukaryotic cell structure? Eukaryotic cell will have a plasma membrane, which is the flexible outer boundary of the cell. On the inside of the cell, acting as its control center, you've got the nucleus. And you will see between the plasma membrane here and the nucleus, we've got a fluid that contains the machinery of the cell, which are the organelles. We call the fluid by itself the cytosol. We call the fluid plus the organelles, along with other inclusions inside the cell. We call all this together is my cytoplasm. So cytoplasm is the intracellular fluid that contains the organelles. All this together is called cytoplasm. Cytosol and organelles. So if you notice on here, this is how the eukaryotic cell is gonna look like. I have a plasma membrane on the outside. I have the cytoplasm that is gonna contain all those organelles, all those different organelles that we're gonna discuss later today. And the control center of the cell is gonna be the nucleus. And if you see, the nucleus has the genetic material, and the genetic material is gonna be condensed in a chromatin form. So the chromatin here is simply is your DNA. At the center of the nucleus, we've got a darker part. We call it the nucleolus. And you will see that you have a double membrane that separates the chromatin and nucleolus from the rest of the cytoplasm. So we've got a double layered membrane surrounding the nucleus. We call this is my nuclear envelope or the nuclear membrane. So starting first with the plasma membrane. Plasma membrane is a bimolecular layer of lipids and proteins that will constantly be changing its appearance. It's a constantly changing fluid mosaic. It plays a dynamic role in cell activity. It also serves as a boundary between the inside of the cell and the outside of the cell. So inside of the cell is what we're going to call the intracellular fluid compartment. And the outside of the cell, the fluid around the cells, this is going to be my extracellular fluid compartment. So again, cell membrane here acts as a boundary between the inside of the cell, the internal structure of the cell, and the outside of the cell, which is my extracellular fluid compartment. So fluid inside the cell is what we call intracellular fluid. Fluid outside of the cell is going to be my extracellular fluid compartment. So looking closer to the structure of this boundary. So I'm magnifying this part of the membrane. How does it look like? So we're looking here at a bilayer of phospholipids. This is how the phospholipids gonna look like. Each phospholipid molecule gonna have a phosphate group, and attached to the phosphate group, we've got fatty acids, 2 fatty acids. So what do we call a structure formed by phosphate and fatty acids? We call this is my phospholipids. So the main component of the plasma membrane is going to be fats. And the most common type of fat that is present in the structure of the cell membrane is going to be my phospholipids. Phospholipids are representing 75% of the total amount of lipids in the plasma membrane. So looking closer at the structure of the phospholipids, we will see that it has a phosphate group, and the phosphate group is polar. What I mean by polar, it has a charge. Phosphate groups are polar. But the fatty acids are non-polar. They don't have a charge. They are neutral. In terms of electricity. So again, phospholipids, they have polar phosphate group that will serve as the head, and non-polar tails, which are the fatty acids. So if you try to put oil in a cup of water, would you be able to mix the oil with water? Or would they be kept separated all the time? Even after you mix them, they will separate. Mix them, they separate, right? So why the oil does separate? Because it's simply non-polar. It doesn't have a charge. And some things that you need to keep in mind, if I am non-polar, I will be water insoluble. I can't be diluted in water. I'm water insoluble. How about if you are using salt? If you put salt, it's a sodium chloride. Sodium chloride, when you mix it with water, the sodium becomes a sodium ion, and the chloride gonna be a negatively charged ion. So what happens, they will become polarized. And some structures that have polarities that are polarized, they are water soluble. So when you mix salt with water, what's going to happen? The salt will become diluted in the water. Why? Because it's water soluble, it's charged. So the same concept on here. So you've got two parts of the phospholipids. Part which is non-polar, which is the fatty acid tails, and part which is polar, which is going to be the phosphate group attached to those fatty acid tails. So where do I have fluid? Where do I have water? I have water, remember, inside the cell. Remember, cytoplasm, located inside is a fluid. It contains a lot of water. Also, around the cells, you have fluid. Remember, you've got extracellular fluid. So you have fluid on both sides of the membrane. So I have here, I have water. Here, I have water. Those are the two sides of the membrane. What we're looking at is the thickness of the line. This is what we're looking at. So you have water here. You have water here. On both sides of the membrane, I've got water. So what's going to happen? Which one loves water? Which one is water soluble? Phosphate heads or the fatty acids? Which one loves water? The heads, phosphate head. So what happens? You see on here, the phosphate head is going to be directed towards the extracellular fluid and towards my intracellular fluid, towards water, inside and outside of the cell membrane. And which ones hate water? They are non-polar fatty acid tails, right? Fatty acid tails, as you see on here, they hate water. So where would they be directed? They would be directed towards the inside of the cell membrane. Again, all this here is the thickness of the cell membrane. So the tails will be directed away from the water because you got water outside of the cell, water inside of the cell. So fatty acids are directed away from this water towards the inside of the thickness of the plasma membrane. Those are my phospholipids. Again, phospholipids is going to be the major component of the cell membrane. They are representing around 75% of the total amount of fat that enters in the structure of the plasma membrane. If we're looking here, so being water insoluble, what do we call something that hates water? It's hydrophobic. What do we call something that loves water? It's hydrophilic. What's phobia? Phobia is fear. Philia is love. All right, so loving water, I'm water soluble, I am hydrophilic. Hating water, I'm afraid of water, I am hydrophobic. So again, what is the structure of the phospholipids? They have hydrophilic heads, which are polar, and hydrophobic fatty acid tails. Heads are directed towards the water. And where is the water? Inside of the cell and outside of the cell, on both sides of the membrane. Where I can escape from water if you are directed towards the inside of the plasma membrane itself. So directing away from the fluid on both sides. Other types of fats that will be present in the structure of my plasma membrane. I've got cholesterol, and the cholesterol here is going to be representing around 20% of the amount of membrane lipids. The cholesterol serves to maintain the membrane stability and fluidity. So try to imagine I'm compressing cells. When I tap like this on my skin, I'm compressing cells. So why those cells did not rupture? Because I'm applying trauma. Why the cells did not rupture? Because simply they are compressible. Their membrane is not rigid. If the membrane of the cells is rigid, what's going to happen if you apply any kind of trauma to them? They will rupture. So it's very important on here to have cholesterol present in the plasma membrane. Why? Because simply this what will give the membrane its fluidity and its stability. Another type of fats that we've got is going to be fats attached to which we've got sugar. So what do we call the sugar attached to lipids or fat? This is going to be my glycolipids. And glycolipids, from the names, they are glyco sugar attached to fats. Glycolipids will share in the formation of something that we call the glycocalyx. What is the glycocalyx? It's formed of glycolipids and proteins that will also be attached to sugar groups like this. Together, the sugar groups attached to proteins and attached to the fats, which are the glycolipids, they will form what we call the glycocalyx. Why do we have a glycocalyx? Why do we have sugar coat around our cells? So what do you think? How your immune system will recognize that this is a bacterial cell and this is your cell? So I will attack the bacteria and I will not be attacking my cardiac muscle cells, for example, or my skeletal muscle cells or my joints. So how the immune system differentiates between your cells and pathogenic cells that get inside? How would it be able to differentiate between the two? What do you think? I check the fingerprint of the cell. And what forms the fingerprint of your cells is going to be the glycolipids and glycoproteins that are forming the glycocalyx. So the sugar groups attached to the bacterial cell membrane are different than yours, which will serve as antigenic markers. So the immune system, when it gets exposed to those markers, it gets excited and starts to attack those pathogenic cells and not your own cells. All right, so this is like a fingerprint to the cell. It helps the cells to recognize one another. So again, we've mentioned that the plasma membrane is formed of lipids proteins, we've got three types of lipids. The most common type, representing 75% of the membrane lipids, is going to be my phospholipids. Second type is going to be the cholesterol, which maintains the membrane fluidity and integrity. And the third type is my glycolipids. And the glycolipids, as you see on here, those are lipids attached to which we've got sugar groups. Why do we have sugar groups? We mentioned that sugar groups are not only attached to lipids, they are also attached to proteins. And all those sugar groups together are what we call the glycocalyx. And all of the sugar groups either attached to lipids or attached to proteins, forming this glycocalyx will be acting as the fingerprint of your cells. All right. So what are the glycolipids? Again, glycolipids are those ones here. Lipids attached to them, we've got sugar groups. You see on here? Those are my glycolipids. But glycoproteins, you see a protein attached to which we've got sugar groups. So what's the difference between a glycoprotein and a glycolipid? Here is sugar attached to lipids, either cholesterol or the phospholipids, compared to the glycoproteins. Those are sugar groups attached to the proteins in the cell membrane. Did this answer your question? Yes. Theodore? Any questions? Any questions? All right, so moving on to the plasma membrane proteins. Proteins are gonna be divided into two groups. All right, so I have two different protein groups in the membrane. So first type is the one that occupies the whole thickness of the membrane. We call those are my integral proteins. Integral proteins. And we've got attached to them much smaller proteins, either directed outside of the cell membrane or inside of the cell membrane. Those are what we call the peripheral proteins. So we've got integral proteins, and here those are my peripheral proteins. Integral proteins are going to be inserted into the membrane, and most of them are transmembrane, means they will be occupying the whole thickness of the cell membrane. They will have many functions, as we're going to discuss later in details. They will be responsible for transport, to transport molecules inside and outside of the cell. They might act as enzymes. They will act as receptors in the cell. Compared to the peripheral proteins, they will be loosely attached to the integral proteins. They will include the filaments on the intracellular surface and the glycoproteins on the extracellular surface. So if we are looking here at the plasma membrane. We can see two types of proteins on here. As you see, this is an integral protein, and this is a peripheral protein. This is another one. Peripheral proteins, on the inside of the surface of the cells, they will be attached to the filaments that are forming the skeleton of the cell. So your cell has a skeleton like yours, but those are formed of protein filaments. And all those protein filaments together are what we call the cytoskeleton, the skeleton of the cell. To the outside, the peripheral proteins that are directed towards the outside, they will have an attachment to the sugar groups. And what do we call those proteins that are attached to sugar groups, sharing in the formation of the glycocalyx? They are my glycoproteins. Glycoproteins. So again, two different types of membrane proteins. We have the transmembrane proteins, and those are my integral proteins. We have loosely attached to those transmembrane proteins, either on the outside surface of the cell membrane or the inside surface of the cell membrane. We've got those peripheral proteins. Functions are going to be to act as enzymes, to allow cell-to-cell links to be formed, to provide the support on the intracellular surface. And we're going to see how they play a very important role in maintaining the cell structure and stability. So our first question for today, for the in-class activities for today, what do you think is going to be the outer covering of the cell is called? What is the outer covering of the cell called? What do you think? Is it the nucleus, the cytoplasm, the plasma membrane? Plasma membrane, right? Plasma membrane. All right, so looking at the different functions of those plasma membrane proteins that we have seen. First, they will be responsible to allow the transport to take place in some cases. So if I want to allow sodium, for example, to travel from outside of the cell to the inside of the cell, this sodium is polarized. You see on here, it's an ion, it has a charge. And if you remember, what is the main component of the cell membrane? What is the main component of the cell membrane? Can you remind me what did we call those ones? Phospholipids, right? Phospholipids. So the sodium is charged. It has a positive charge on here. Can I pass through the plasma membrane, through the phospholipid bilayer? Can I just pass through the between the molecules of the phospholipid bilayer? What do you think? Do I have a problem with the phosphate group? It's charged. It's water soluble, like me. I don't have much of a problem. The problem is with the tails. Why the tails are going to be a problem for the sodium? Because the tails are hydrophobic. They hate water. They hate water. They can't allow a water-soluble molecule to, or ion here to travel. Same for glucose. Can I allow the glucose in? Glucose has charge. Would I be able to pass through the phospholipid bilayer? I, I am hydrophilic, and the fatty acid tails on here are hydrophobic. So they won't allow me in. So how would I allow sodium and glucose to the inside of my cells? I need something that allow their transport. And those are actually going to be plasma proteins. So some plasma proteins will act as channels for the sodium to be able to travel to the inside of the cell if needed. Some of the plasma proteins will have more complex shape, larger structure to allow larger molecules like the glucose to travel to the inside of the cell. And we call those ones that are transporting the glucose, those are my GLUT or glucose transporters. So you see on here, the first function of the plasma membrane proteins will be to allow the membrane transport to take place, especially for the hydrophilic structures. Why the hydrophilic structures can't pass by easily through the plasma membrane? Because simply the plasma membrane has fatty acid tails. So here it's going to be hydrophobic. I have fat. I can't allow something that is water soluble to travel in. You can't mix water with oil, remember. So what allows the cell to get sodium, to get glucose, and many other stuff from outside in or from inside out is going to be the presence of those plasma membrane proteins that will be responsible to allow the transport to take place. Some plasma membrane proteins will be acting as surface receptors. If, for example, I did just eat, let's say a donut. So what's going to happen? I will break down this donut. And I will digest it, and I will form the type of sugars that I'm able to absorb, which is the glucose. Now the glucose is present in my small intestine. It needs to be absorbed. So when it gets absorbed to my blood, what's going to happen to my blood sugar level after I absorb, after I eat this donut? It goes up, it increases, right? So my blood sugar level will go up as I eat this donut. Why do we eat? Why do we eat? Why do we get glucose? For our cells to be able to break it down and produce energy. Exactly. So do you want the cells to be getting this glucose in all the time, whether you did eat or not? Or let's say another example, another cell, just one specific cell. So if it was extra glucose, I will store this energy. So can you remind me, what cell did we study that stores energy? Fat cells. So let's say those are my fat cells. Would you like the fat cells to keep pulling the glucose all the time from your blood if you don't eat or you do eat in both cases? What do you think? No. You want only the fat cells to pull the glucose whenever you have high blood sugar for the extra sugar to be stored. So what we're gonna do, we're gonna be relying on an endocrine gland in the pancreas. Pancreas here acts as an endocrine gland. It senses the increase of the blood sugar level and it responds by releasing insulin. So when I see insulin means I have high blood sugar. And what is the insulin function? The insulin function is to allow this glucose to move to the inside of the cell. The insulin function is not just to lower down the blood sugar level, it allows the sugar to move to the inside of the cell. This is my main function. And as a result, the blood sugar will drop. Blood sugar level drops as a result of the intake of the glucose to the inside of the cells. All right. So how the cell will know that it needs to uptake the glucose? It needs to allow the glucose in. How the insulin will be acting on the cell in order for it to uptake the glucose. Remember, in order to uptake the glucose, I needed to have the GLUT present, so glucose transporters. So normally I don't have this glucose transporter on the surface of the cell. So how would I know that there is an increase in the glucose and it's time now for me to form the GLUT to uptake this glucose? I wait for a signal, and the signal here for me is going to be the insulin. And the insulin can't get to the inside of the cell, it's still polarized. So what will I do? I have receptors, and those are proteins on the surface of the cell, plasma membrane proteins that will act as receptors for insulin, and they will be sending the signal to the inside of the cell. For the cell to do what? For the cell to build up the glucose transporter, which is another plasma protein that will allow the glucose in. So this is just to give you an idea about how surface membrane proteins can act as receptors. So you've got a signal, I've got a hormone, for example, outside like the insulin in this case, and I want to respond to this insulin. How would I know that there is insulin, which means I have high blood sugar? I need to have receptors for this insulin. This insulin can't pass to the inside of the cell to tell the cell that you've got a lot of glucose outside. So I need something that will bind to the insulin and transmits the signal to the inside of the cell. And in this case, those going to be the insulin receptors. And who again acts as insulin receptor? It's going to be my plasma membrane protein. Is this concept clear? Is this concept clear? Any questions? Questions? All right, so moving on to another function of the plasma membrane protein, and this is going to be serving as the attachment of the cytoskeleton on the inside of the cell and the extracellular matrix. So you see on here, what do you think? Are your cells free to move around, generally speaking? So if this is an epithelial cell, for example, like this, can it leave its place and move around to the deeper tissues? What do you think? Or is it kept in its place? It's kept in its place, right? You have surface proteins that will attach it to the extracellular matrix and also going to be attaching the cytoskeleton, the filaments on the inside of the cell. I have surface membrane proteins that will be attaching the cell to the surrounding matrix, the outside, and the inside of the cell membrane, the inside of the cell also going to be attached. So yes, there is a basement membrane on here, but it's not only the limiting factor for those cells to not move deeper. So what, for example, what do we check for an adenoma or an epithelial cancer? What do we check under the microscope to differentiate between a benign tumor and a malignant tumor if we see an invasion of the of the basement membrane? If the malignant cells are proliferating on here without passing to the inside, to the deeper tissues through the basement membrane, this is going to be a localized tumor, for example. So what keeps it like this? What keeps it attached to the surrounding, including the basement membrane and the extracellular matrix? Again, is the presence of those plasma proteins. So again, plasma membrane proteins are going to be serving as an attachment to the cytoskeleton on the inside of the cell, as well as the extracellular matrix. Other function of the plasma membrane is going to be to enhance and speeds up chemical reactions by acting as enzymes. And we have thousands and thousands of examples of those chemical reactions that will be taking place in our cells. All right, so I don't want to discuss complicated examples at this point. Just remember that those plasma membrane proteins can speed up the rate of chemical reactions acting as enzymes. All of them gonna have enzymatic activity. All right, we're gonna discuss an example of this later on when we get to chapter 16. Also, the cells are going to be attached to one another, and this is going to be the intercellular joining. So what keeps the cells attached together is the plasma membrane proteins on both sides. So one of them is negatively charged, one of them is positively charged, for example. So they would be kept attracted and attached to one another. Cell-to-cell recognition, remember what's going to be allowing the cell to recognize other cells? is the presence of the sugar groups attached to either the fat or the protein. And again, all the sugar groups together, either attached to proteins or fats, this is what we've called again the glycocalyx. So again, glycocalyx on here is formed by both glycolipids and glycoproteins. And again, what is the importance on here? It allows cell to cell recognition. Moving on to how the cells are attached to one another. So cells might be attached by tight junctions. And if I look closer on here to what is a tight junction, you'll see that you have protein filaments that are stitching the two cell membranes to one another, as if I'm stitching the membrane. So it travels from one membrane to the other, from one membrane to the other. So this will allow the cells to stick together, not only sticking together, but also will allow you to prevent anything from passing between the cells. And where do you want, in what kind of tissue do you want that very strong attachment between the cells? Where do you want to have tight junctions between your cells? What do you think? Where would you like to have tight junctions? In which type of tissue? According to the functions that we've discussed. Epithelium. Why epithelium? Why epithelium? Because epithelium, what was the function of the epithelium? Can you remind me again? Epithelium was responsible for formation of the boundaries. So if I'm forming a boundary, either a covering or a lining, would I like something to travel between the cells? I like something to travel between the cells. If I'm the boundary. No, you need to travel through the cell to allow the absorption to take place. Remember, in the epithelium, I don't want to have, in most of the cases, something that is passing between the cells. I want to make sure that everything that travels from one side to the other is traveling through the cell to be monitored and regulated by these cells. All right, I don't want to have stuff passing between the cells. So that's why we're going to have tight junctions in a lot of epithelial tissues. Another type of membrane junctions and cell junctions is going to be the desmosomes, rivets, and those are going to be acting as attachments by having a plate on each cell. So this is cell number 1 and this is cell number 2. So you see on each cell, inside each cell, we've got a plate. This plate is going to be attached to the cytoskeleton of the cell, and this plate is going to be attached to the cytoskeleton of the other cell. And those two are going to be attached by sugar attached to proteins, by glycoproteins. So here, what keeps the cells attached together is the glycoprotein that is attached to the plate in both cells. What do we call the linker glycoproteins on here? Those are my Cadherence. Cadherence. So what do we call this whole structure that allows the cells to be attached to one another? Those are my desmosomes. Moving on to another type of membrane junctions, which going to be the gap junctions. Gap junctions, from the name, what do you think? Are you attaching the cells close to one another, or they have a lot of space in between? What do you think? From the name, they are called gap junctions. So what do you think? Do you have a space between the cells, or the cells are attached together? And remember, here, those are junctions. They are attachments. So cells, yes, the cells are attached to one another. They are attached to one another. Don't get confused by gap. Having a gap doesn't mean that I have a space between the cells. All right? The cells are still attached to one another, but what attaches the cells, if you see on here, are protein channels like this, which will keep the cells connected. This is what we mean by gap junctions. Again, gap junction, do I have a space between the cells attached by gap junctions? They are junctions. No, we don't have a space. But what do I mean by gap junctions is the presence of those channels that will be connecting them to one another. And why would I need gap junctions? Why would I need gap junctions? To allow the movement of signals freely between the cells. So I've got an electrical signal on here, let's say, and I want this signal to travel to all the cells in the same time. Like in what situation? Like, for example, if I'm looking at the cardiac muscle cells. So let's imagine on here, this is my heart like this. This is my ventricle, actually. This is my left ventricle. This is my right ventricle. And the blood is supposed to be pumped from the right side towards your lungs. And it's going to be pumped from the left side to be distributed to all the different systemic body organs. All right, so don't worry about the details on here. The signal will reach the heart, the cardiac muscle cells at the apex of the heart, like this. These are the first cells that get excited. Would you like this electrical signal to keep exciting few cells, one cell at a time, few cells, few cells, few cells, few cells, few cells, few cells? So try to imagine those cells, when excited, gonna do what? They will contract. So let's say, would you like those cells to contract first, and then those ones, 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 and then those ones? Or would you like them all to contract in the same time? What do you think? Would you like each few cells to contract? for them to contract in a sequence like this. So you have a delay in the conduction of the signal. So the signal is taking time to reach all the cells. Or would you like all of them to contract in the same time? All of them to contract in the same time. Why? Because you understand that in order for me to squeeze the blood out, I need to create an effective contraction, and this can be reached only if I contract all the cells at the same time. So how would I conduct the signal to all the cardiac muscle cells in the same time is the presence of gap junctions. So the gap junctions will allow the signal to travel freely between the cells, allowing the cellular coordination, allowing free transmission of the signals without having delay in the electrical conduction. And this is what I want in the heart. And this is what we're going to see again later on in smooth muscles. So in the type of smooth muscles, we're going to have gap junctions to allow them to coordinate the muscle movement, like for example, in your intestines or stomach. So you don't want to have part of your intestine contracting and the other part is not. So you want to have a coordinated contraction in your intestines, and this is going to be allowed by having a free transmission of the signals that will allow them to contract by having again what kind of junctions between those cells. So this is going to be my gap junctions. So major example on here, where do I keep gap junctions? Where do I keep gap junctions? Cardiac muscles and smooth muscle cells. All cardiac muscle cells are attached by gap junctions with one part of the heart, only one part of the heart that has fewer gap junctions. And smooth muscles, we have one type of smooth muscles that will have a lot of gap junctions. So if you notice, I hit again here, cells are attached to one another, but I have channels connecting those cells to one another. And we call those are the connexon. Connexon, those are the channels connecting the adjacent cells to one another, allowing the free conduction of the signal. But we can have a 10 minutes break. We come back, start our discussion of the membrane transport. Any questions before we leave for a break? Any questions?